Hello everyone. Today we are going to start a new topic, cell cycle. We will discuss about different stages of cell cycle, how it progresses and how different stages are regulated. What is cell cycle? All the eukaryotic cells have life cycle and that is known as cell cycle. It has two periods. One is interphase. This is the period of no apparent division. Here, no division in the cell is seen. And the other is M phase. This is the division phase. It can be mitosis or meiosis. Earlier it was thought that interphase is the resting stage between two consecutive division. But cell spends most of the lifespan in the interphase. It is the phase in which intense biological activity takes place. Cell grows and double its size. Growth of cell takes place and all the molecular units like chromosome and all, the duplication takes place. And in the M phase, that is the division phase, the cell division is seen, but actually it is the separation of already duplicated molecular units. If we see the eukaryotic cell cycle, there are four successive stages: G1 phase, S phase, G2 phase and M phase. G1 is the gap between the end of mitosis phase and the start of the S phase. In the G1 phase, the preparation takes place for the DNA synthesis or DNA replication. All the enzymes and the proteins that are required for the DNA replication takes place in the G1 phase. In the S phase, replication of the DNA takes place. G2, it is the gap between the end of the S phase and the start of the mitosis phase. Among all the phases, the most important is the G1 phase, because in the G1 phase only the cell decides whether it has to go in the new cell cycle or it has to arrest the cell cycle. And the cell in the G1 phase decides this depending upon all the internal and external conditions. If the internal and external conditions are favorable, it continues to the cell cycle. If it is not favorable, then cell exits from the or it withdraws itself from the cell cycle and enters into the G0 stage, and then the cell cycle is arrested. Cell remains in the G0 stage for days, months, years, weeks, depending upon the condition, and when the condition changes, then it again resumes the proliferation or it again enters the cell cycle. Some of the cells like nerve cells and muscle cells, these remain in the G0 or the resting stage throughout their life. Some, like the liver cells, after a long period, these can enter into the cell cycle. In the cell cycle, G1 phase, S phase and G2 phase are together known as interphase. And this is of longer duration of S phase, G2 phase and M phase in all the cells of an organism is constant. That is, in any particular organism, all the cells have the fixed duration of S phase, G2 phase and M phase, but G1 phase is variable. In eukaryotes, diploid cell has DNA content of 2C in the G1 phase. In the S phase, duplication of the DNA takes place, so the DNA content is 4C in the G2 phase. Again it enters the mitosis and 2 daughter cells are produced, and these daughter cells enters the G1 phase, and these have the DNA content 2C, and in this way the cycle continues. We have seen that in the cell cycle there are 4 successive stages which progresses in sequence. After G1, S phase takes place, then after the S phase G2 takes place, and then after the G2 phase the M phase takes place. There are certain engines that helps in the progression of the cell cycle. This is actually protein complex. It has 2 subunits. One is cyclin, which is the regulatory component. And the second is CDK, which is catalytic component. It catalyzes the phosphorylation. Cyclin, the name is given because it undergoes cycle of synthesis and degradation. When the cyclin is required, it undergoes synthesis, and when it is not required, it is degraded. And in this way, the synthesis and degradation continues in cycle. CDK is cyclin dependent kinase. This CDK, its activity is regulated by the cyclin, and this cyclin only decides appropriate protein for the phosphorylation. So we have seen that cyclin undergoes synthesis and degradation, but CDK is present throughout the cycle. When the CDK combines with the cyclin, then it is active. For the cell cycle, there are series of cyclin CDK complexes. These are known as G1 cyclin CDK complex, S phase cyclin CDK complex, and M phase mitotic cyclin CDK complex. This G1 cyclin, S phase cyclin and M phase cyclin only appear during the G1 phase, S phase and M phase, but CDK is always present. In yeast, there is only one CDK which binds with different cyclins at different phases. But in vertebrates, there are more than one CDK which binds with different cyclins in different phases. So we can conclude that only specific cyclin CDK complex are present or are active during the specific phase of the cell cycle. That means G1 cyclin CDK complex is active only during the G1 phase. S phase cyclin CDK complex is active during the S phase, and so on. So there is certain regulatory mechanism which control their activity. What are these regulatory mechanisms? Let us see how the activity of cyclin CDK complex is regulated. Their activity is regulated by three ways: cyclin expression and degradation, phosphorylation and dephosphorylation, and third is binding of CDK inhibitor proteins, or CKIs in short. So let us discuss them one by one. First is cyclin expression and degradation. Cyclin expression and degradation. Let us understand with an example. Mitotic cyclin CDK complex, also known as MPF, M phase promoting factor or maturation promoting factor. It is synthesized in G2 and S phase. It results in the chromosomal condensation, breakdown of nuclear envelope, assembly of mitotic spindles, and then chromosomal alignment along metaphase plate. This mitotic cyclin CDK complex also activates anaphase promoting complex, APC. This APC is ubiquitin ligase. This causes the proteolytic degradation of anaphase inhibitor. And when anaphase inhibitor is degraded, then it results in the inactivation of protein which binds the two sister chromatids. This is two sister chromatids, this is bind by the protein. When this protein that is inactivated, then these are no more bind and these separated. This results in the onset of anaphase, and after the anaphase telophase takes place, and later on it has to exit from mitosis. And how this exit takes place? This occurs with the help of this anaphase promoting complex. It is inactive. When it binds with CDC20, it becomes active. This active APC CDC20 complex then results in the proteolytic degradation of this mitotic cyclin. Mitotic cyclin is degraded and then the cell exits from the mitosis. The events that takes place during this is chromosomal decondensation takes place. Then the nuclear envelope is formed around the daughter nuclei. After that, cytokinesis takes place and two daughter cells are formed. So we can conclude that specific cyclin is expressed for the specific stage of cell cycle. And once this stage is completed, then the cyclin undergoes degradation. Then starts the next stage of the cell cycle with another set of cyclin CDK complex. In this way, cell cycle progresses in only one direction and it is irreversible. Activity of cyclin CDK complex is also regulated by phosphorylation and dephosphorylation reaction. We can understand with the example of the S form, which is the yeast. Here the cyclin component is CDC13 and CDK component is CDK1. These combines and forms the cyclin CDK complex, which is inactive. Now V1, which is protein tyrosine kinase, phosphorylates tyrosine residue present in the CDK. This is inhibitory phosphate. So the cyclin CDK complex is inactive. Now CAC, which is CDK activating kinase, phosphorylates threonine residue present near the active site of CDK. So it is activating phosphate. But this cyclin CDK complex is still inactive because of the presence of this inhibitory phosphate. When this cyclin CDK complex is required in the cell cycle, then activity of V1 decreases and activity of CDC25 increases. This is protein phosphatase. This then removes inhibitory phosphate from CDK. Now this cyclin CDK complex has only activating phosphate and this cyclin CDK complex is now active. So this phosphorylation and dephosphorylation regulates the activity of cyclin CDK complex on the basis of their requirement in cell cycle. Cyclin CDK complex activity is also regulated by binding of CDK inhibitor protein. CDK inhibitor proteins bind with cyclin CDK complex. When CDK inhibitor protein binds with cyclin CDK complex becomes inactive. Some of the CDK inhibitor proteins are P27, P21 and P16. These control G1 or S phase of cell cycle. Cell cycle progresses in controlled manner or regulated manner. It is important for the cell cycle to proceed in the favorable condition, but it doesn't proceed during the unfavorable condition. So how it is decided whether the cell has to progress the cell cycle or it has to arrest the cell cycle? For this, there are several checkpoints present in the different stages of the cell cycle. Among them, three major checkpoints are G1 checkpoint, that is present at towards the end of the G1 phase, G2 checkpoint, which is present towards the end of the G2 phase, and metaphase checkpoint, that is present in the M phase. At these checkpoints, cells decides whether the condition, internal or external condition, is favorable for the cell cycle to proceed or not. So let us see these checkpoints one by one. First is the G1 checkpoint. It is the main checkpoint. It checks various factors like whether the size of the cell is enough to undergo division, then whether all the nutrients that is required for the cell growth and division is available or not, whether it is getting positive signals for the division like growth factors, etc., and it checks whether DNA is damaged or not. If all the conditions are favorable, then the cell cycle progresses through the G1 phase and then enters the S phase for the duplication of the DNA. And if these conditions are not favorable, then the cell cycle is arrested. It enters the G0 stage, which is known as the resting stage, and remain in the G0 stage till the favorable conditions return. Second is the G2 checkpoint, and this is towards the end of the G2 phase. It checks whether the duplication of DNA is complete or not. It also check whether DNA is damaged. And if any problem is there, then the cell cycle pause at this point and tries to complete the DNA replication if it is incomplete, or if any damage is present, it tries to repair. This is because if it is not checked at this place, then after the cell division, the daughter cells that is obtained will have the problem in their DNA, whether it will be incomplete or any damage will be present, and this can lead to problems like cancer, etc. So it is important to check at this place and try to resolve this problem. If the problem is beyond repair, then the cell undergoes apoptosis, which is known as programmed cell death. Third is metaphase checkpoint, also known as spindle checkpoint. At this point, cell checks whether the chromosomes are properly attached to the spindle fibers in the metaphase plate. This is metaphase plate, this is chromosome. There are two sister chromatids attached. And this is the spindle fibers to which chromosomes are attached. So it checks whether the chromosomes are properly attached to the spindle fibers, because after metaphase comes anaphase, and in the anaphase there is separation of the sister chromatids. If these chromosomes are not properly attached to the spindle fibers, then the proper separation of sister chromatids will not take place, and we know that anaphase is irreversible. Role of RB protein in cell cycle regulation. E2F, it is the transcription factor which binds with specific sequence of DNA and helps in the transcription of cyclin A gene. Gene whose product is required to promote S phase. So when E2F is active, then it transcribes the gene and gives the product, and cell enters the S phase and cell cycle continues. In the normal cells, it is important to regulate the activity of this E2F in order to pause the cell cycle and see if all the conditions are favorable and there is no problem in the continuation of the cell cycle. This is controlled by or regulated by the RB protein. This RB protein has the affinity for E2F. When it binds with it, it becomes inactive, and so now no transcription of the gene takes place. Cyclin D CDK4 complex or cyclin D CDK6 complex phosphorylates RB protein. This phosphorylated RB protein then does not have the affinity for E2F. This happens in the mid-G1 before the checkpoint or the restriction point, as a result of which RB protein does not binds with E2F and the cell enters the S phase. RB protein is the product of RB gene. It is called tumor suppressor gene because when it is inactivated by the mutation, it results in the tumor. This happens in retinoblastoma. Retinoblastoma is childhood tumor of retina. Here, both the sets of RB gene is inactive, and the protein which is transcribed from this RB gene is inactive and it does not have the ability to control the cell cycle, as a result of which the cell cycle goes uncontrolled and it results in the tumor of retina. Role of p53 in cell cycle regulation. p53 gene is known as tumor suppressor gene and its product is p53 protein, which results in the cell cycle arrest at G1 and G2. In the normal condition, when no damaged DNA is present, then the p53 protein is very unstable. It undergoes proteolytic degradation by the MDM2, which is ubiquitin ligase. But when damaged DNA is present, then this damaged DNA results in the activation of ATM serine kinase. This ATM serine kinase then phosphodilates p53 protein. Now, this MDM2 cannot bind with the phosphorylated p53 protein. Hence, the degradation of p53 doesn't take place. Level of p53 protein increases. This p53 protein is the transcription factor for p21 gene and it helps in the synthesis of p21 protein. This p21 protein is CDK inhibitor. It then binds with the S-phase CDK. S-phase CDK is active. When p21, which is CDK inhibitor, binds with it, then it becomes inactive. Then the cell cycle are arrested at the G1. Damaged DNA is repaired and after it is repaired, the cell cycle progresses. Sometimes the damage of the DNA is beyond repair. In that case, cell undergoes apoptosis. This is programmed cell death. If p53 gene is lost or mutated, then cell cycle is not arrested at the G1, hence the repair of DNA does not take place, as a result of which cell with the mutated genome undergoes proliferation and it becomes cancerous. So this is all for cell cycle. If you like this video, please hit the like button, share it and subscribe my channel. In the next video, we will learn about mitosis. Thank you.